the last part of the process of making a protein is called translation. And what happens in translation is you take that information and you translate it into an actual protein. Okay, and that's done on a ribosome, and that's why there's a picture of a ribosome here, although it doesn't look like that. And basically, you're transferring from your codons into, which are nucleic acids, three nucleic acids together, into an actual sequence of amino acids. Those a sequence of amino acids is just another name for a protein or a polypeptide. All these are the same name, same thing. Amino acids are just the individual monomers, units of the protein or a polypeptide. All right, so again, I'm going to have a little animation of this broken down, like not as fast as that first one. Kind of like I had an animation of the other one broken down. And I'll show you that in a second. But first, what happens is the messenger RNA is going to travel outside of the nucleus. One of those questions asked in the worksheet was, where does transcription take place? The fact is transcription takes place inside the nucleus. This stuff travels outside the nucleus to a ribosome. Where are the ribosomes usually located? You remember from biology class? There was a rough ER and smooth ER. What did ER stand for? Endoplasmic reticulum. Yeah, it's a great name. Always like that name. It's one of those names you don't forget. First, the messenger mRNA, messenger RNA, travels out of the nucleus into a ribosome, and that consists of really two units. You may notice them kind of fit together in the previous animation. Okay, and there are binding sites. You may have noticed that as well. You're going to see it again in the thing. And here's I thought there was another line coming up there. Hit that light for me. Notice that? See how she did that, Stone? See that? Just turn the light off, just like that. All right.
Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has two binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
Hello? Yeah, well, that's period. Isn't it next period? No, I'm sorry. Take it back. Okay. All right. So I'm going to ask you, because I'm going to give you some actual codons, sequence of them, and I'm going to ask you to translate them into what the amino acids should be that as they go. But basically, and notice it only had 2 sites, but there's actually 3. It's not really important. The point is, as it goes into each of these sites, it slides to the next one, and then it breaks off and after it joins the peptides at the top. You also notice that there are start and stop sequences, start and stop codons. Thionine is a start codon, and there are several stop codons. So when I give you a sequence, you should be able to, tomorrow we're going to be doing this, and make it, you're going to actually, some of those are going to correspond to specific traits. All right, so it's just a neat little worksheet. Well, let's talk about this now and breaking it down here. A complementary transfer RNA molecule binds to the exposed codon. Now, those amino acids that are at the other end, they get put right next to each other. So they are able to join. Once they get really close to each other, it's a natural chemical reaction that's going to happen between an acid and a base, or it's a carboxylic acid, and an amino. The amino part is the basic. So those 2 guys get together, and they form a compound. They split water off. So you're going to join these 2 guys when they get close together, and grow a long chain. If the messenger RNA, like this guy has AUG, which is a start sequence, then you're going to expect a transfer RNA is going to have UAC, which correspond to methionine. And you have these same pictures in your thing too. All right, the ribosome helps that peptide bond get formed, and then it breaks off. All right. And moves over to a different site, and then finally leaves the molecule, and allows the other guy to come in and slide down. So you keep growing these chains. Like I said, they can be hundreds of amino acids long. They keep growing on this ribosome, keep putting them out. You may remember again from the video of this chain of snaking out of the ribosome, this red chain that was making the hemoglobin in the previous one, not this one here, but the one before that yesterday, last period. Eventually, you will reach what's called a stop codon. All right. When it reaches the stop codon, the ribosome breaks apart, and a long chain of amino acids breaks off, and it usually goes and does something else as well. It usually gets folded up into a 3-dimensional structure that will go out into the cell and do whatever function that cell's supposed to be doing. So again, after that guy breaks off, you're going to polypeptide eventually folds into a protein and performs whatever function it's supposed to be doing. Notice this, we have a long chain now of polypeptides, much longer than just 4 or 5 like I showed you there, but you get the idea. And this chain is not just one straight long chain. Like I said, it folds upon itself and eventually forms a three-dimensional structure that is whatever this guy I'm going to show you a picture of is hemoglobin. But he has, in order to be functional, he has to be able to do that. As a matter of fact, one of the things that happens if you were to you could make a protein non-functional is with changes in pH, because they will not fold properly, and that has to be regulated in the body as well. And these are just examples of proteins in your body. Any enzymes of protein, hemoglobin is the one there's a picture of, insulin, okay? Like there's a picture of hemoglobin folded up. I think you have that on the bottom there. The way it folds because of hydrogen bonds between neighboring amino acids. And that's what makes them fold into the shape that makes them work. You know hydrogen bonds are weaker than the covalent bonds actually holding the guy together, actually holding the backbone of this protein together. All right, and the last thing, just one last thing here. Amino acids. You notice all those amino acids are out in the cell. You watch the animations. They're getting picked up by these transfer RNA. Your body's able to make many of them, but it can't make 8 of them. 8 of the 20 amino acids are called essential amino acids. They have to be ingested. You have to eat them. Those of you who are vegetarians, for example, have to be careful. I can't just eat rice. If I just eat rice, I am not going to get every essential amino acid because not every essential amino acid is in rice. Okay? Can't just eat beans. If I just eat beans, I won't get every essential amino acid because not everyone's in it. If I eat meat, I will. If meat, dairy products actually have all of the essential amino acids in them. So people who are vegetarians do have to watch what they eat more. And that's why a lot of times people who are vegetarians have pairings of rice and beans, for example, because with the two, you'll get all the essential amino acids. They often are paired together because you have to eat them to be able to make everything. All right. Now, I'm going to give you another worksheet, but before I do, on the back of this one that you have, I want you to do this problem, okay? And what it is, and I'm going to try to make sure everybody understands this before we go on. Okay, before we do that, we need Kirsten to have one more job on the lights here.
With computer animation, we can enter the cell.
This is basically just an overall picture of what's going on here. All right, again, started with one, I'm going to end with one. You can see basically what's happening in protein synthesis, but also a lot of the functions of the cell.
To view this remarkable system at work.
With the DNA.
After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line.
Animation you can see. These are the codons. These are the anticodons of the transfer RNA. This is the messenger RNA. And here down here are each individual amino acid. So this is basically the process. All right, we broke it down at each step of it. How this was made, that's transcription, and then how these are being added together, that's translation. But this is probably the best animation for all of them put together.
Builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
That's basically it. So that's pretty impressive. That's happening all the time in your cells. Okay, so you want to copy this onto the back of your sheet. You don't have to copy the whole question. Just copy this. The sequence, this sequence here. Leave room underneath it for what I want you to write there. There I would want you to write the complementary mRNA sequence that would have formed from this, and then underneath that, you're going to write the amino acids that will be formed from that. And I'll talk about that in a second. But just so basically just copy this TAC, CGT, whatever, all right? And directly underneath that, leave room for the mRNA sequence. And then underneath that, for the amino acid sequence. And you could use abbreviations because I'm going to give you this chart that has the abbreviations and what the codons actually correspond to.